There is a world beneath your feet that most human beings will never see. It has no sunlight. It has no seasons. It has no weather. The temperature stays the same every day of every year. A cold and perfect constancy that makes the surface world, with its storms and droughts and blazing summers, seem almost hysterical by comparison. Sound moves differently here. Smell is everything. Time, in the way that surface creatures experience it, barely exists. And yet this world is full of life, not the sparse, scraggly life of extreme environments, the kind that clings to the edges of habitability and barely survives. Real life. Diverse, specialized, extraordinary life that has been evolving in complete darkness for millions of years, producing animals so different from their surface relatives that when scientists first encountered them, they sometimes refused to believe they were looking at members of the same planet's fauna. Blind fish that navigate obstacle courses more precisely than sighted fish can. Spiders that have retained vestigial eyes they cannot use, but that still go through the developmental motions of building them as if the genome remembers what the animal has forgotten. Insects with antennae so sensitive they can detect the chemical signature of a single molecule in a cubic meter of cave air. Crustaceans that have been sealed in the same underground lake since before the dinosaurs went extinct, cut off from the surface world by geological events that happened so long ago, they predate the rise of the mammals, evolving in isolation into forms that have no close relatives anywhere on Earth. Salamanders that live for 100 years without ever seeing light, bacteria that derive their energy not from sunlight, but from the chemical reactions of rock and water, building ecosystems from scratch in the complete absence of the photosynthesis that powers virtually every other ecosystem on the planet's surface. Caves that are essentially alien worlds, operating on entirely different energetic principles, populated by creatures that evolution has reshaped with a precision and completeness that makes them, in the truest biological sense, citizens of a different Earth. This is the world of cave life, and it is stranger, richer, and more scientifically important than almost anything you will encounter in the more celebrated wild places of the surface world. To understand why caves produce such extraordinary life, you first have to understand what caves actually are, because the popular image of a cave, a dark hole in a hillside leading to a chamber with stalactites and bats, captures almost nothing of the reality of cave systems as geological and biological environments. Caves are not simply gaps in rock. They are the result of processes that operate over millions of years, driven by the chemistry of water and stone, creating systems of passages and chambers that can extend for hundreds of kilometers in 3 dimensions, dropping from near the surface to depths of more than 2 kilometers below ground, connecting lakes and rivers and aquifers and air-filled passages in architectures of extraordinary complexity. The most extensive cave systems are formed in limestone, a sedimentary rock composed primarily of calcium carbonate, the same material that makes up the shells of marine organisms. Limestone dissolves slowly in slightly acidic water, and the slightly acidic water that falls as rain and percolates through soil picks up carbon dioxide, forming weak carbon dioxide, which is then carried underground by gravity along fractures and bedding planes in the rock. Over tens of thousands of years, this slow chemical dissolution widens the fractures into passages. The passages grow, converge, and interconnect. Underground rivers form, carving their own channels through the rock with the combined forces of dissolution and mechanical erosion. Chambers form where passages intersect or where the rock is structurally weaker and collapses The whole system grows and changes on timescales that dwarf human civilization, with some passages dating back tens of millions of years and representing geological histories of extraordinary complexity. The result of this process, played out across millions of cave systems on every continent except Antarctica, is a global underground network of passages and chambers whose total extent is essentially unknown. New cave systems are discovered every year. Every significant spelunking expedition to a known system returns with maps of previously unmapped passages. The best estimates suggest that the total linear extent of explored cave passages on Earth is somewhere in the hundreds of thousands of kilometers, with a much greater extent remaining unexplored or undiscovered. Mammoth Cave in Kentucky, the longest known cave system on Earth, has more than 660 kilometers of surveyed passages and continues to yield new extensions with each exploration season. The La Chiguila Cave in New Mexico, discovered in its deeper sections only in 1986, contains more than 230 kilometers of passages and reaches depths of more than half a kilometer below the surface, with a geological history and a biological community that have been described by researchers who have studied them as unlike anything else on Earth. The Mobile Cave in Romania, sealed from the surface for at least 5 million years, contains an ecosystem powered entirely by chemosynthesis, by the metabolic activities of bacteria that derive energy from the oxidation of hydrogen sulfide, rising from the rocks below with no dependence whatsoever on sunlight or on organic matter from the surface. When scientists first entered Mobile Cave in 1986, they found 48 species, 33 of them entirely new to science, all living in a closed system that had been evolving in complete isolation since before modern humans existed. The Vrykolakas Cave in Greece, the Kubra Cave in Georgia, the deepest known cave on Earth, reaching more than 2 kilometers below the surface. The Irwang-Dong Cave System in China, which contains a climate system so complex that it generates its own clouds inside the mountain, a permanent mist that hangs in the vast chambers of the cave in the complete absence of sunlight. Driven by temperature differentials between different parts of the cave system, these are not simply geological curiosities. They are biological laboratories of extraordinary power, places where evolution has been running controlled experiments in adaptation to extreme environments for millions of years, producing results that illuminate some of the most fundamental questions in evolutionary biology. The organisms that live in caves are called troglobites, a word derived from the Greek for cave and life, and they are among the most intensely studied subjects in evolutionary biology precisely because they offer something that is almost impossible to find elsewhere: living examples of evolution in action, animals that can be compared directly to their surface-dwelling relatives, allowing scientists to measure exactly what has changed, what has been lost, what has been gained, and over what timescale when a population is removed from the pressures of the surface world and placed in the utterly different pressures of permanent darkness. What they have found, studying these animals, has rewritten our understanding of how evolution works, what drives the loss of complex traits, how nervous systems adapt to the absence of a sense they evolved to use, how metabolism reorganizes when energy is severely limited, and how isolation produces novelty in biological systems that have been cut off from gene flow with the wider world. The story of cave life is, in this sense, more than a natural history story. It is a story about the mechanisms of evolution itself, told through the bodies and behaviors of animals that have been shaped by those mechanisms more completely and more readably than almost any other organisms on Earth. And it begins, as so many evolutionary stories do, with a single critical event, the moment when a surface animal first enters the dark and decides, or is forced to stay. The colonization of caves by surface animals is not a single historical event. It has happened independently, thousands of times, in cave systems on every continent, involving hundreds of different animal lineages, over time spans ranging from a few thousand years to tens of millions of years. Each colonization is a separate evolutionary experiment, beginning with a different starting organism, running in a different cave environment, and producing a different outcome. But the initial process is remarkably consistent across these different experiments. It begins with a surface population that lives near the entrance of a cave system and that uses the cave entrance as a refuge from predators, as a shelter from extreme temperature, or as a hunting ground for the insects and other invertebrates that accumulate around cave entrances, where organic material from the surface, leaves, dead animals, plant debris, is concentrated by water flow. Over time, individuals that are willing to venture deeper into the cave gain access to resources that are not available to individuals that stay near the entrance. The interior of the cave is relatively predator-free compared to the surface because most surface predators cannot navigate in complete darkness. It may offer stable temperatures during seasonal extremes on the surface. It may provide access to prey species that have already made the transition to cave life and that are available in the interior but not at the entrance. Natural selection favors individuals that can exploit these resources, which means individuals that can navigate in reduced or absent light, that can find food without using vision, that can survive on the sparse and unpredictable energy resources of the cave interior. Over many generations, these selective pressures accumulate, pushing the population toward a suite of adaptations that is so characteristic, it has been given its own name, the cave syndrome. The cave syndrome includes both gains and losses. The gains are sensory, enhanced chemoreception, the ability to detect chemical cues at extremely low concentrations, enhanced mechanoreception, the ability to detect vibrations and pressure waves in air and water that provide information about the environment without requiring vision, enhanced tactile sensitivity. In aquatic cave animals, the elaboration of the lateral line system, the network of pressure-sensitive cells along the sides of fish and amphibians that detects water movement to a degree of sensitivity far beyond what is seen in surface relatives. The losses are equally characteristic: the reduction and eventual disappearance of eyes, the loss of pigmentation, the reduction of circadian rhythms, the internal biological clocks that track the cycle of day and night, which serve no purpose in an environment where there is no day and no night. The modification of metabolism to deal with the feast and famine energy availability of cave environments, the extension of lifespan and the reduction of reproductive rate. This constellation of traits, the suite of adaptations to cave life, appears again and again across unrelated lineages in fish, in Mexico, and in fish in China, in spiders in the Canary Islands, and in spiders in the caves of New Zealand, in beetles in Slovenia, and in beetles in Australia. It is one of the most striking examples of convergent evolution in all of biology, the independent invention of the same solutions to the same problems by organisms that share nothing but the environment they have adapted to. The convergence is so consistent and so precise that early cave biologists, confronted with the same traits appearing in completely unrelated animals, were sometimes confused about the degree of evolutionary relationship between the cave animals they were studying. How could a blind, depigmented, long-lived cavefish from a Mexican river cave look so similar to a blind, depigmented, long-lived cavefish from a Chinese cave if they were not closely related? The answer, of course, is that they are not closely related at all. They simply face the same problems, and evolution has found the same answers. Understanding why these specific answers are found so consistently, why darkness produces blindness and depigmentation, and enhanced non-visual senses, and extended lifespans so reliably, regardless of the lineage, requires understanding the economics of biological structures in environments where energy is the limiting resource. The cave environment is, energetically speaking, desperately poor. The primary source of energy in cave ecosystems is organic matter that washes or falls into the cave from the surface. Plant debris carried by underground rivers, insect bodies, blown into cave entrances, insect bodies, blown into cave entrances, the guano of bats and cave-roosting birds that feed on the surface and deposit their waste underground, dissolved organic carbon carried by percolating groundwater. All of these sources are sparse and unpredictable. The total energy available per unit volume of cave space is a tiny fraction of what is available in any surface ecosystem, even the most impoverished desert or polar tundra. In this context, every biological structure is an energy cost. Every organ the animal builds and maintains requires energy to construct during development and energy to repair and maintain throughout life. Eyes are particularly expensive organs. The vertebrate eye is a complex structure requiring the development and maintenance of multiple cell types, including the highly metabolically active photoreceptor cells that must regenerate their light-sensitive pigments continuously, the neural processing cells of the retina, the lens cells, and the intricate network of blood vessels needed to supply oxygen and nutrients to all of these cells. In a surface animal, this investment pays for itself many times over in the survival advantage that vision provides, the ability to detect predators, to find food, to navigate, to communicate visually, but in complete darkness, eyes provide none of these advantages. A perfectly functional eye in a totally dark cave is as useless as a solar panel in a lightless room. Natural selection in a cave environment therefore ceases to maintain the investment in eyes. Mutations that reduce eye development or that divert the cellular and energetic resources previously allocated to eye development toward other uses are no longer selected against. In a surface environment, such mutations would be strongly deleterious, reducing visual acuity and compromising survival. In a cave environment, they are neutral at worst and potentially beneficial if they redirect resources toward more useful structures. Over many generations, the accumulation of mutations that reduce eye development proceeds without check from natural selection, and eyes degenerate. The process is not instantaneous. It typically takes tens of thousands to hundreds of thousands of years for eyes to be completely lost, depending on the generation time of the organism and the rate at which mutations accumulate. But in the geological timescales of cave occupation, complete eye loss is a reliable outcome in animals that have been cave dwellers for long enough. The same logic applies to pigmentation. Melanin, the primary pigment of animal skin and scales, is metabolically expensive to produce and serves its primary function on the surface as protection against ultraviolet radiation from sunlight. In a cave, there is no ultraviolet radiation. Pigment production is pure cost with zero benefit. Mutations reducing melanin production are neutral in cave environments and accumulate over time, producing the characteristic ghostly pallor of long-established cave animals. The loss of circadian rhythms follows the same pattern. The biological clocks that time daily cycles of activity, metabolism, and physiology are costly to maintain and serve no function in an environment with no light-dark cycle to track. They degrade over evolutionary time in cave populations, producing animals whose activity patterns are no longer tied to a 24-hour clock, which is not only metabolically economical, but also functionally useful in an environment where food availability is patchy and unpredictable rather than rhythmically organized around a daily cycle. These reductive changes, the loss of eyes, pigment, and circadian rhythms, were for a long time the primary focus of cave biology, partly because they are the most visually striking features of cave animals and partly because they seemed to offer a clean test case for evolutionary theory. But the constructive changes, the gains in non-visual sensory capability, the metabolic reorganizations, the life history shifts toward longer lives and lower reproductive rates are equally important and in some ways more interesting because they represent positive adaptations rather than simple relaxations of selection. The elaboration of non-visual senses in cave animals is among the most impressive examples of sensory evolution documented in any group of organisms. In the cavefish of Mexico, the Texas blind salamander, the olm of European caves, and dozens of other well-studied cave animals, the loss of visual processing has been accompanied by an expansion of the brain regions devoted to processing non-visual sensory information, a structural change in the brain that reflects a fundamental reorganization of sensory priorities. The Mexican blind cavefish, Astyanax mexicanus, has become the model organism for cave biology in the same way that the fruit fly Drosophila is the model organism for genetics. It has a surface-dwelling form and a cave-dwelling form that are members of the same species and can interbreed when brought together in the laboratory, allowing direct genetic comparison between the two forms. This comparison has revealed the specific genetic changes responsible for the cave syndrome traits with a precision that is not possible in cave organisms that have no living surface relatives. The lateral line of the cave form of Astyanax is dramatically more elaborate than that of the surface form. The number of sensory hair cell clusters, called neuromasts, on the head and body of the cavefish is significantly greater, and the neuromasts themselves are larger, containing more hair cells each and projecting further from the body surface. These modifications make the cavefish's lateral line exquisitely sensitive to the pressure waves created by objects in its environment, essentially giving it a three-dimensional image of its surroundings through water movement rather than through light. The cavefish can detect an object the size of a coin from a distance of several body lengths using lateral line information alone. It navigates around obstacles in complete darkness with an accuracy that exceeds the performance of sighted fish in comparable tasks under normal lighting. It detects prey movement at distances and under conditions of water turbidity that would render a surface fish completely blind to the same prey item. This is not merely the compensation of one sense for the loss of another. It is the development of a genuinely superior sensory system for the specific conditions of the cave environment. The blind cavefish is not a deficient version of the surface fish. It is a specialized version optimized for its specific habitat in ways that make it more capable in that habitat than its sighted relatives could ever be. The blind cavefish of Mexico has been studied more intensively than perhaps any other cave animal on Earth, and the insights that research has produced go far beyond the specific biology of a single fish species. They have illuminated the genetic architecture of complex traits, the mechanisms by which the same mutations can produce different effects in different genetic backgrounds, and the evolutionary dynamics of populations adapting to novel environments. The research began in earnest in the 1960s when geneticists realized that Astyanax mexicanus offered a unique opportunity, a species with multiple independent cave populations, each derived from a different surface ancestor and each at a different stage of evolutionary transition to cave life, living alongside a surface population that remained morphologically similar to the common ancestor. This arrangement made it possible to study not just what had changed, but how it had changed by comparing the cave populations of different ages and degrees of differentiation. What the genetic research revealed was surprising in several ways. The first surprise was the number of genes involved in the cave syndrome. Rather than being controlled by a small number of master regulatory genes that switch eye development on or off, the reduction of eyes in cave Astyanax turned out to be controlled by many genes, each contributing a small effect whose cumulative action over many generations gradually reduced eye size and function. This polygenic architecture of cave adaptations is now understood to be the general rule rather than the exception. The cave syndrome is not the result of a few dramatic mutations, but of the gradual accumulation of many small genetic changes, each neutral or slightly beneficial in the cave environment, that add up over time to produce the dramatic phenotypic differences between cave and surface populations. The second surprise was the relationship between eye loss and jaw development. One of the most consistent findings across different cave Astyanax populations was that the genes contributing to eye reduction also contributed to changes in jaw and taste bud development, with many cave populations showing enhanced development of taste buds on the lips and jaws compared to surface fish. This suggested that the genetic pathways controlling eye development and jaw development were deeply interconnected, so that selecting for reduced eyes simultaneously produced enhanced taste bud development through shared developmental pathways. This is a beautiful example of a principle called antagonistic pleiotropy in reverse. Rather than a single gene having beneficial effects in one trait and detrimental effects in another, here the loss of one trait vision appears to be genetically coupled to the gain of another, enhanced chemoreception through shared developmental pathways. The evolution of cave adaptations is therefore not simply a story of loss, but of developmental reallocation, of the redirection of developmental resources from structures that are useless in the cave to structures that are beneficial. The genetic comparison between cave and surface Astyanax has also revealed something surprising about the repeatability of evolution. The different cave populations of Astyanax were colonized independently at different times by different surface fish, yet they have converged on similar suites of cave adaptations, including reduced eyes, enhanced lateral line, increased taste bud number, and metabolic changes. When researchers compared the specific genetic changes underlying these similar adaptations in different cave populations, they found significant overlap. Many of the same genes and even the same specific mutations had appeared independently in different cave populations, producing the same phenotypic outcome through the same genetic mechanism. This parallel evolution at the molecular level, the repeated appearance of the same mutations in the same genes in independent cave populations, is one of the most striking demonstrations of evolutionary determinism ever documented. It suggests that the genetic paths available for adaptation to cave environments are constrained, that certain genetic changes are so beneficial in cave conditions that natural selection reliably finds them wherever they are available in the standing genetic variation of cave colonizing populations. The implications of this finding extend beyond cave biology. If evolution can be predictable at the genetic level, If certain mutations are so reliably beneficial in certain environments that they appear repeatedly across independent populations, then evolution is less random than the classical view suggested. It has attractors, evolutionary outcomes that are pulled toward by the landscape of fitness in specific environments, and understanding those attractors allows us to make predictions about the direction of evolution that were not previously possible. The cave environment, with its clean before and after comparison between cave and surface populations, has been the laboratory in which this insight was developed. But the principle it illustrates applies to evolution everywhere. Not all cave organisms are fish. In fact, the diversity of life that has colonized caves is extraordinary, spanning virtually every major group of animals and many groups of microorganisms. Understanding that diversity requires a survey that moves through the different types of cave environments and the different organisms that have adapted to them. Because the variety of cave habitats is itself remarkable, ranging from the completely aquatic environments of flooded cave passages to the dry, dusty chambers of desert caves. From the fog-filled tropical caves of Southeast Asia to the ice caves of volcanic islands, each with its own physical and chemical characteristics and its own community of adapted life. The aquatic cave environment, the world of flooded passages and underground lakes and rivers, is home to some of the most dramatically adapted cave organisms on Earth. Water is a more forgiving medium than air for animals that have lost their eyes, because water carries both chemical and pressure wave information far more effectively than air, allowing sightless animals to build detailed three-dimensional maps of their surroundings through non-visual senses. The aquatic cave fauna is consequently diverse and often highly specialized, with species that have evolved in isolation for millions of years in underground waters that were sealed from the surface by geological events in the deep past. The olm, Proteus anguinus, is perhaps the most famous of all cave animals, and it deserves its reputation. It is a cave salamander, a member of the family Protei that inhabits the underground waters of caves throughout the Dinaric Alps of southeastern Europe in a range that spans parts of Slovenia, Croatia, Bosnia-Herzegovina, and Italy. It is pale, almost translucent, with a pinkish tinge caused by the hemoglobin visible through its unpigmented skin. It has vestigial eyes covered by skin. It has 3 pairs of feathery external gills that it retains throughout its life, having never completed the metamorphosis from larval to adult form that most salamanders undergo, a condition called neoteny. The olm can grow to more than 30 centimeters in length. It feeds on invertebrates in the cave water, detecting prey through chemoreception and possibly mechanoreception rather than vision. It breathes through its gills, though it can also take air at the water surface when oxygen levels in the cave water drop. But the most extraordinary thing about the olm is not its appearance, it is its lifespan. Olms can live for more than 100 years. Research published in 2010 by a team led by Jan Voiteron of the Université Lyon, based on mark recapture studies of wild olms over several years, estimated the maximum lifespan of the olm at approximately 102 years, with a median lifespan of around 68 years. These are not laboratory animals kept under optimal conditions. These are wild animals living in the dark, cold, food-poor environment of a cave system. A lifespan of more than a century in a relatively small amphibian is extraordinary by any standard. The longest-lived amphibians on the surface rarely exceed 20 to 30 years. The olm's longevity is not an accident or an anomaly. It is an adaptation to the specific conditions of its cave environment, part of the life history syndrome that cave animals evolve in response to the sparse and unpredictable food availability of caves. The logic of this life history shift is grounded in evolutionary theory. When adult mortality is low, as it tends to be in cave environments, where there are few predators and where the physical environment is stable and predictable, natural selection favors delaying reproduction and investing more heavily in body maintenance because the expected future reproductive value of a surviving adult is high. Conversely, when food is scarce and unpredictable, as it is in caves, investing heavily in a large clutch of offspring during a single breeding event is a risky strategy because the offspring may not find enough food to survive. Producing fewer offspring more carefully and surviving to reproduce again next year and the year after that is a more reliable strategy in an environment where survival is possible but food is scarce. The result is the characteristic cave life history: slow development, late maturity, small clutch sizes, high parental investment per offspring, and exceptional longevity. The olm represents the extreme end of this continuum among vertebrates, but similar patterns appear across cave faunas worldwide. The olm's sensory capabilities are as remarkable as its lifespan. In the complete absence of functional vision, it has developed chemical senses of extraordinary sensitivity. The concentration of chemoreceptors on its snout is far higher than in any surface salamander, and behavioral studies have shown that olms can detect chemical signals in the cave water at concentrations that are essentially undetectable by standard chemical analysis equipment. They can locate prey items buried in sediment several centimeters below the cave floor, using chemical gradients to pinpoint the exact location of hidden food. They also appear to have a magnetic sense. Studies published in the 2010 S demonstrated that olms could orient consistently relative to the Earth's magnetic field in featureless water environments, a capability that would be extremely useful in the complex, three-dimensional maze of an underground cave system where other directional cues are absent. The magnetic sense of the olm is not fully understood at the neurobiological level, but its behavioral reality has been demonstrated in controlled experiments, and it represents another example of the expansion of non-visual sensory capability in cave animals. The underground lakes of the Dinaric Karst, where olms live, are also home to communities of cave-adapted invertebrates that have been evolving in isolation for periods that dwarf even the olm's impressive lineage history. Isopods, small crustaceans related to pill bugs and woodlice, have been found in these cave systems in forms so different from their surface relatives that they represent not just species, but entire genera and even families found nowhere else. Cave amphipods, small shrimp-like crustaceans, have radiated in the underground waters of the Dinaric Karst into dozens of species, many of them blind and depigmented, filling ecological niches in the cave ecosystem with a diversity that rivals the most species-rich surface freshwater communities. The isolation of these cave crustacean communities dates back to tectonic events that restructured the drainage systems of the Balkans millions of years ago, sealing populations of surface crustaceans in underground waters that subsequently became disconnected from each other and from the surface world. Each isolated population evolved independently, accumulating the cave syndrome traits and diversifying within its specific cave environment, producing the extraordinary diversity of cave crustaceans that makes the Dinaric Karst one of the most intensively studied regions in cave biology. The geological history that isolated these populations is itself an important part of the story. Caves are not static environments. They evolve over geological time, just as the organisms within them evolve. Cave passages that were once flooded may drain as the water table drops. Passages that were once dry may flood as tectonic uplift changes drainage patterns. Entrances that once connected cave systems to the surface may be blocked by sediment deposition or collapse. And entire cave systems may be isolated from neighboring systems by geological events that cut off the underground water connections that previously linked them. Each of these geological events is a potential speciation event for cave organisms. When a cave system is divided by a geological barrier, the populations on either side of the barrier are no longer in contact, no longer exchanging genes, and begin to evolve independently. If the separation lasts long enough, the populations accumulate enough genetic differences to become reproductively isolated, which is the standard definition of separate species. The caves of the Dinaric Karst have been a laboratory for this process for millions of years, and the result is a cave fauna of exceptional diversity and scientific value. The crustaceans, insects, spiders, and other invertebrates of these caves represent an irreplaceable evolutionary archive, a record of millions of years of isolated evolution that cannot be recovered if it is lost. The threat to this archive is real. Cave systems worldwide are under increasing pressure from human activities. Groundwater extraction for agriculture and industry lowers water tables, draining the aquatic habitats that cave species depend on. Agricultural runoff carrying pesticides and fertilizers contaminates cave water through percolation from the surface. Quarrying and construction in karst landscapes physically destroys cave systems. Cave tourism, if not carefully managed, can disrupt the delicate physical and biological equilibrium of cave ecosystems through the introduction of non-native organisms, the alteration of cave air currents, and the simple physical disturbance of organisms and structures that have existed in stable conditions for millions of years. The conservation biology of cave species is complicated by several factors that do not apply to surface organisms. Cave species are often so specialized that they can survive only in the specific cave system where they evolved, and sometimes only in specific parts of that system. A cave invertebrate that has evolved in the cold, oxygen-poor water of a deep underground lake may not be able to survive in any other water body on Earth. If that lake is contaminated or drained, the species that depend on it are gone. Not regionally extirpated, not locally depleted, but globally extinct. The invisibility of cave species compounds the problem. Surface biodiversity is visible. Forests, reefs, and grasslands can be surveyed from the air, monitored by satellite, and assessed for their conservation status in ways that generate public attention and political will. Cave biodiversity is hidden. Most people have no idea that the groundwater beneath their community may harbor unique species found nowhere else on Earth. Most political jurisdictions have no legal framework that adequately protects underground biological communities because those communities are not included in the baseline surveys that inform conservation. The International Union for Conservation of Nature estimates that a higher proportion of cave species are threatened with extinction than any other major ecological category. The reasons are clear: extreme specialization, tiny ranges, invisibility to conservation planning processes, and vulnerability to the specific human activities that impact cave environments. Cave species are being lost before they are found, as geological surveys sometimes discover the remnants of cave ecosystems, empty shells, fossilized bones, dried exoskeletons that were once living communities in caves that were destroyed or contaminated before anyone knew they were there. Among the most biologically astonishing cave environments on Earth Mobile Cave in Romania stands alone, not because of the spectacular formations that draw tourists to other caves, not because of dramatic underground rivers or vast chambers, but because of what it taught us about the fundamental nature of life itself when it was first entered by a small team of Romanian scientists in 1986. Mobile Cave is located near the town of Mangalia on the Black Sea coast of Romania. It had been sealed from the surface for an estimated 5 to 5.5 million years, cut off by geological processes that blocked all connections to the outside world. The water inside the cave is warm, around 21 degrees Celsius, fed by a geothermal source that has maintained stable conditions through the entire period of isolation. The cave contains a series of air-filled chambers above a lake of slightly toxic water rich in hydrogen sulfide, ammonia, and carbon dioxide gases that would be lethal to most surface animals. When the Romanian scientists entered, they expected to find a cave essentially devoid of life. What they found instead changed the field of biology. The cave contained a thriving ecosystem, an abundant and diverse community of organisms living in conditions that should have made life impossible, sustained by an energy source that had nothing to do with sunlight. The basis of the ecosystem was a mat of bacteria covering the surface of the cave water, bacteria that derived their energy not from photosynthesis, but from the chemical oxidation of hydrogen sulfide, a process called chemosynthesis. These bacteria were the primary producers of the mobile cave ecosystem, the organisms at the base of the food chain, playing the role that plants and algae play in surface ecosystems, not through capturing sunlight, but through capturing the chemical energy of reduced sulfur compounds rising from the rocks below. Above the bacterial mat, a community of invertebrates fed on the bacteria. Specialized water lice, amphipods, snails, and worms, all blind, all depigmented, all representing species found nowhere else on Earth. Above these primary consumers were carnivores, cave spiders, pseudoscorpions, and other predatory invertebrates that fed on the herbivores. The entire community, with its multiple trophic levels and its complex web of interactions, was sustained entirely by the chemical energy of the geothermal system, with no input of organic matter from the surface and no dependence on sunlight anywhere in the chain. The discovery of the mobile cave ecosystem was not just a discovery of new species. It was a fundamental revision of our understanding of the energetic basis of life. Before mobile, the concept that had dominated biology since the development of the theory of photosynthesis was that all complex ecosystems ultimately depend on sunlight. That photosynthesis by plants, algae, and cyanobacteria is the universal foundation on which all animal life rests, because photosynthesis converts the energy of sunlight into the organic compounds that fuel everything else. Deep-sea hydrothermal vent ecosystems, discovered in 1977, had already challenged this view by demonstrating that chemototrophic bacteria could support complex animal communities in the complete absence of sunlight. But the deep sea is a difficult environment to study, and its physical extremes of pressure and temperature seem to limit its relevance as a model for broader biological principles. Mobile Cave demonstrated that chemosynthesis-based ecosystems were not restricted to the deep sea. They could exist in the underground environment, accessible from the land surface, sustained by geothermal, rather than hydrothermal chemistry, in conditions that were physically much less extreme than the deep sea. And they had been doing so for millions of years, evolving in complete isolation, developing communities that were biologically rich and ecologically complex in the complete absence of any input from the sunlit world. The organisms of Mobile Cave have been the subject of intensive study since 1986, and each expedition has added to the picture of extraordinary evolutionary specialization that characterizes this community. The cave spiders of Mobile, members of the genus Hadzihalopithasma and several other genera, have evolved hunting strategies specifically adapted to the foggy, chemically unusual atmosphere of the cave airspace above the water, detecting prey by vibration rather than vision in conditions of complete darkness and high humidity. The cave water lice, members of the genus Armadillidium, have evolved digestive systems capable of processing the sulfur-rich bacterial mats that form their primary food source, with enzymatic capabilities not found in their surface relatives. The cave leeches have evolved prey detection systems based on chemical gradients rather than the visual or vibration-based detection used by surface leeches. The isolation of Mobile Cave has been so complete and so prolonged that researchers estimate the average cave organism has had between 5 and 10 million years to diverge from its surface relatives. Over this timescale, the genetic distances between cave and surface forms are substantial, in some cases placing the cave organisms in different genera from any surface relative, representing evolutionary distances comparable to those between species that last shared a common ancestor before the emergence of the grasslands that now cover much of the Earth's surface. The bacteria of Mobile Cave deserve special attention because they represent a completely novel biochemistry compared to anything found in sunlit surface environments. The hydrogen sulfide oxidizing bacteria that form the base of the Mobile ecosystem have been found to possess enzymatic pathways for processing sulfur compounds that have no equivalent in surface organisms. They produce novel compounds, tolerate chemical conditions that would inhibit or kill the enzymes of surface organisms, and maintain the unusual metabolic capabilities needed to derive energy from the specific mixture of reduced gases present in the Mobile geothermal system. The detailed biochemistry of these bacteria is still being characterized. Each expedition to Mobile returns with samples that, when analyzed in the laboratory, yield new information about the metabolic innovations that 5 million years of isolated chemosynthetic evolution have produced. Some of the enzymes discovered in Mobile Cave bacteria have potential applications in industrial chemistry and in the treatment of industrial waste streams containing sulfur compounds, because they can process those compounds under conditions where conventional catalysts fail. The Mobile Cave ecosystem also provides what is, in effect, a 5 million year natural experiment in what happens to evolutionary dynamics when a community is sealed from outside genetic input. In surface ecosystems, species are continuously exposed to gene flow from neighboring populations, to the immigration of new species from outside, to the periodic disruption of mass extinction and recolonization events. These inputs continuously inject new genetic variation into populations and communities, providing the raw material for evolutionary change. In Mobile Cave, none of these inputs have been available for 5 million years. The gene pool of every organism in the cave is exactly and only what was present when the cave was sealed, plus the mutations that have accumulated since. The evolutionary trajectories of the cave organisms have been shaped entirely by mutation, genetic drift, and natural selection operating within a fixed genetic starting point, with no external genetic input of any kind. The result is, in evolutionary terms, a set of lineages that have gone as far as their initial genetic endowment, modified by millions of years of mutation, can take them. Their adaptations are, in this sense, the deepest possible expression of what natural selection can do with a limited genetic toolkit over a very long time. And what it has done in 5 million years is produce organisms that are so thoroughly adapted to their specific environment that they are now incapable of surviving anywhere else on Earth. The complete dependence of mobile organisms on their specific cave environment is both the measure of their evolutionary success and the source of their vulnerability. They are, in biological terms, masterpieces. They are also, in conservation terms, extremely fragile. The seismic activity of the Black Sea region, the drawing of groundwater from the karst aquifer system by agriculture and industry in the Mangalia area, the potential for contamination of the geothermal water system that feeds the cave. Any of these could disrupt or destroy the mobile ecosystem before it has been fully described and understood. The conservation status of Mobile Cave is now protected by Romanian law, and access is strictly controlled. Only a handful of scientists have entered the cave since its discovery, and each expedition is carefully planned to minimize the introduction of surface organisms or the disturbance of the cave's physical and chemical equilibrium. The cave is, in this sense, treated not as a tourist attraction or a scientific resource to be exploited, but as a biological archive of irreplaceable scientific value to be protected with the same care as a unique manuscript or an irreplaceable work of art. The caves of the world contain hundreds of mobile-like systems, smaller and less dramatically isolated, but each containing communities of organisms that have been evolving in their specific underground environment for timescales ranging from thousands to millions of years. The caves of the Canary Islands, formed by volcanic activity and subsequently colonized by surface invertebrates that have adapted to cave conditions over millions of years, contain communities of blind cave beetles, spiders, and crustaceans that have radiated into dozens of species. The lava tube caves of the Azores, Hawaii, and the Canary Islands host parallel cave faunas that have evolved independently in geologically similar environments, providing some of the clearest examples of convergent evolution in cave biology. The caves of New Zealand contain communities of cave invertebrates that have been isolated since the geological separation of New Zealand from Gondwana more than 65 million years ago, meaning that some cave lineages in New Zealand have been evolving in underground environments for longer than the dinosaurs have been extinct. The Weddas, large orthopteran insects related to crickets, have cave-adapted forms in New Zealand that have evolved reduced wings, enhanced tactile senses, and life history traits characteristic of the cave syndrome. The cave weta, Pakairama, is found in caves throughout the North and South Islands of New Zealand, navigating the cave environment with an ability that seems impossible for an insect, but that its extraordinarily sensitive antennae, capable of detecting chemical and mechanical signals in the dark cave air with remarkable precision, makes entirely comprehensible. The cave glowworms of New Zealand and Australia represent one of the most visually spectacular of all cave phenomena. The New Zealand cave glowworm, Arachnocampa luminosa, is not in fact a worm, but the larva of a small fungus gnat. It produces bioluminescent light from a specialized organ at the end of its abdomen, using a biochemical reaction similar to but independently evolved from the bioluminescence of fireflies and deep-sea organisms. The larva hangs sticky threads from the ceiling of the cave to catch small flying insects attracted to the light, a predatory strategy of elegant simplicity that turns the cave ceiling into a glowing, trap-laden sky. In the caves of the Waitomo region of New Zealand, and in similar caves in Australia, These larvae cover the ceilings in numbers that produce a remarkable effect, a constellation of blue-green points of light in the darkness of the cave ceiling, as if the night sky has been brought underground and its stars rearranged in a pattern determined not by cosmology, but by the distribution of sticky hunting threads. The visual effect is genuinely extraordinary, and it has made the Waitomo Caves one of the most visited natural attractions in New Zealand. The bioluminescence of the cave glowworm is itself a fascinating evolutionary story. Bioluminescence has evolved independently more than 40 times in different animal lineages, each time using the same basic biochemical strategy. The enzyme luciferase catalyzing the oxidation of a substrate called luciferin to produce light, but with different luciferase and luciferin molecules derived from different ancestral proteins, reflecting the independent invention of the same biochemical solution in different genetic contexts. The bioluminescent system appears to have been derived from an ancestral system used by related surface species for a different purpose, and subsequently modified and elaborated in the cave environment, where its utility as a prey-attracting device provided strong selection for its enhancement. The story of bioluminescence in caves is part of a broader story about the use of light in environments where there is no external light source. Bioluminescence in caves is used not just by glowworms, but by several other cave organisms in different parts of the world, each using light for different purposes in the deep darkness of the cave environment. Certain cave millipedes produce bioluminescent secretions that may function as a warning signal to predators, advertising the presence of toxic chemical defenses without the visual context that would normally make such a warning effective. Cave fungi produce bioluminescent fruiting bodies that may attract the insects needed to disperse their spores in an environment where the wind dispersal used by surface fungi is not available. The use of light in an environment devoid of external light is, in a sense, the ultimate expression of evolutionary creativity. The invention from scratch of a light source in a world where light does not exist, because the presence of light, even in small quantities, provides biological advantages that can be exploited by an organism capable of producing it. It is evolution making light in the dark. The air-breathing cave fauna is no less diverse or remarkable than the aquatic. The terrestrial cave environment, the world of dry and semi-dry cave passages and chambers, is home to a community of invertebrates that have adapted to conditions of complete darkness, low and stable temperature, high humidity, limited food, and the specific architectural complexity of cave passages and surfaces. Cave beetles, members of the family Carabidae and several other families, have undergone dramatic evolutionary change in cave environments on every continent where caves contain beetles. The cave carabid beetles of Europe, studied intensively by cave biologists since the 19th century, have evolved eyes ranging from the full compound eye of surface relatives to the complete absence of any eye structure, with every intermediate stage represented by different species in different caves, providing a natural series illustrating the progressive reduction of the eye over evolutionary time. Their legs have lengthened relative to body size, an adaptation that appears in cave beetles globally, and that is thought to improve their ability to navigate the complex three-dimensional surface of cave rock in complete darkness. Their antennae have become significantly longer, expanding the sensory reach of the mechanoreceptors and chemoreceptors with which they probe the cave environment. The cave carabid beetles of the genus Hedisia in the caves of the Balkans, studied in detail by researchers from the Slovenian Academy of Sciences, represent some of the most specialized cave invertebrates in the world. They have evolved to navigate the thin films of water that coat cave surfaces in highly humid cave passages, a microhabitat that surface beetles cannot exploit, using a body plan that is essentially flat and elongated, with splayed legs that maintain contact with the thin water film on vertical or even overhanging cave surfaces. Their food is the bacteria and algae that grow in these thin water films, a resource that no surface beetle exploits, making the Hadiesia beetles not just cave specialists, but microhabitat specialists within the cave environment. The specialization of cave organisms to microhabitats within the cave is a consistent feature of the most diverse and long-established cave faunas. The caves of Slovenia, Croatia, and the surrounding Dinaric karst region contain cave organisms specialized to different zones of the cave environment: the entrance zone, the twilight zone, the cave interior, the deep passages below the water table, the thin films of water on cave surfaces, the guano deposits of cave-dwelling bats, the percolating water falling from above in zones of active dripping. Each of these microhabitats contains its own specialized community of organisms, many of them endemic to a single cave or a small cluster of connected caves. This ecological specialization within caves mirrors the ecological specialization seen in surface ecosystems, where different species occupy different niche within the same landscape. But in cave ecosystems, the niche differentiation is even more extreme because the physical and chemical heterogeneity of the cave environment is unusually high relative to the small spatial scale of the system, and because the long periods of isolation have allowed endemic species to accumulate in numbers that would be impossible in surface environments subject to more frequent colonization and extinction events. The spiders of the world's cave systems represent one of the most diverse and least understood components of the global cave fauna. Spiders have colonized caves independently on every continent, and in many cave systems they are the dominant terrestrial predators, occupying the role that snakes and lizards fill in surface ecosystems. Cave spiders have evolved a range of adaptations to cave life that parallel those seen in other cave arthropods: eye reduction, leg elongation, enhanced chemoreception and mechanoreception, slowed metabolism, and extended lifespan. But spiders have also evolved cave-specific behaviors that are not seen in other cave arthropod groups, behaviors that reflect the unique capabilities and constraints of the arachnid body plan in a cave environment. The cave spiders of the genus Tetrabolamidae in the caves of Southeast Asia represent some of the most extreme eye reduction seen in any spider group. Some species retain vestigial eyes that can no longer form images, but that still contain photoreceptor cells capable of detecting light. These vestigial photoreceptors appear to be used for circadian rhythm regulation, not for vision. The spiders use the small amount of light that penetrates even deep cave environments from distant entrance zones to synchronize their internal clocks with the natural light-dark cycle. This observation has important implications for understanding why eye reduction in cave animals is incomplete in many species. The standard account predicts that in complete darkness, natural selection will favor the complete elimination of eyes, since eyes serve no function and consume energy. But the cave Tetrabolamidae demonstrate that eyes may serve residual functions even in deep caves, where the absolute darkness of the cave interior is not quite as absolute as it seems to human eyes adapted to daylight. The very small amounts of light present, even in deep caves, may be detectable by highly sensitive photoreceptors. And if those photoreceptors provide any adaptive advantage, such as circadian rhythm entrainment, selection will maintain them even when they can no longer form images. This nuance in eye reduction the maintenance of photoreceptor function after the loss of image-forming capability has been documented in cave animals from multiple groups and represents an important refinement of the simple picture of complete eye loss in cave-adapted organisms. The cave harvestmen, members of the order Opiliones, have colonized cave systems across North America, Europe, and Asia, and represent another group in which the cave syndrome has been documented in remarkable detail. The cave harvestmen of the Appalachian cave systems in the eastern United States have been studied intensively by researchers from universities in Virginia, Kentucky, and Tennessee, and the results have illuminated both the ecology of cave harvestmen communities and the evolutionary processes that have shaped them. The most remarkable aspect of Appalachian cave harvestman biology is the degree of population genetic structure that has developed within individual cave systems, different sections of the same cave, separated by passages that are physically accessible, but that harvestmen rarely traverse, have been found to harbor genetically distinct subpopulations that show significant allele frequency differences at multiple genetic loci. In some cases, the genetic differences between populations in different sections of the same cave are comparable to the differences between populations in entirely separate cave systems, suggesting that the physical architecture of the cave creates effective barriers to gene flow even within a single connected system. This extreme population genetic structure has important implications for the evolutionary dynamics of cave organisms. It means that even within the spatially confined environment of a single cave, evolution can proceed quasi-independently in different subpopulations, potentially leading to local adaptation to the specific physical and chemical conditions of different cave sections. And it means that the number of evolutionarily independent lineages in a single cave system may be much larger than the number of morphologically distinct species, because genetic differentiation can substantially precede morphological differentiation. The cave crickets of the New World, members of the family Rhaphidophoridae, represent another group of cave arthropods that have been studied extensively, both for their cave adaptations and for the insights they provide into the ecological dynamics of cave communities. Cave crickets are unusual among cave animals in that they are not permanent cave residents. They spend the daylight hours roosting in caves and emerge at night to feed on the surface, returning to the cave before dawn. They are what cave biologists call trogloxenes, cave dwellers rather than cave obligates, and they play a role in cave ecosystems that is disproportionate to their biomass. The importance of cave crickets to cave ecosystem function is primarily through their role as vectors of organic matter from the surface to the cave interior. When cave crickets feed on the surface and then return to the cave to roost, they bring with them in their digestive systems the organic material they have consumed. What they deposit in the cave as excrement represents a significant subsidy of surface-derived energy to the cave ecosystem. The guano of cave crickets and the dead bodies of cave crickets that die in the cave are primary food sources for the cave invertebrate community, supporting populations of scavenging beetles, mites, flies, and other detritivores that in turn support the cave predator community. Studies of cave cricket guano ecology have shown that the spatial distribution of cave cricket roosting sites within a cave determines the spatial distribution of the guano-dependent invertebrate community. Caves where crickets roost throughout the interior support a more evenly distributed invertebrate fauna than caves where crickets roost only near the entrance, demonstrating that the energy subsidy provided by cave crickets is a key determinant of the structure and distribution of the cave community. The dependence of many cave invertebrate communities on energy subsidies from the surface is a recurring theme in cave ecology. Very few cave ecosystems are truly self-sufficient in the sense of being energetically independent of the surface world. The chemosynthesis-based ecosystems like Mobile Cave are the exception rather than the rule. Most cave ecosystems receive a significant fraction of their energy from organic matter originating at the surface and the channels through which that organic matter enters the cave. Bat guano, cricket, excrement, plant debris carried by underground rivers, animal carcasses washed or fallen into cave entrances determine the spatial structure and the biological composition of the cave community. This dependence on surface energy subsidies makes cave ecosystems particularly vulnerable to changes in the surface environment. If the surface above a cave is converted from forest to agriculture, the quality and quantity of organic matter entering the cave changes. The invertebrates and microorganisms that decompose leaf litter in cave soils are replaced by bacteria adapted to decomposing agricultural waste. And the cave invertebrate community that depends on forest leaf litter as a food source may decline. If the bat colony that deposits guano in a cave is disturbed or eliminated, the guano-dependent invertebrate community loses its primary energy source and collapses. The connection between the surface and the cave is, in this sense, not just a geological and hydrological connection, but a biological one, a flow of energy and organic matter that links the surface ecosystem to the cave ecosystem in ways that make the health of the cave dependent on the health of the surface above it. The bats that roost in caves are among the most important links in this surface-cave connection. Bats are highly mobile animals that can travel many kilometers from their cave roosts to feeding areas on the surface, consuming large quantities of insects or fruit and depositing the waste products of that consumption in the cave as guano. A large bat colony can deposit tons of guano per year in a single cave, representing an enormous energy subsidy that can support cave invertebrate communities of remarkable diversity and abundance. The guano communities of tropical caves, particularly in Southeast Asia and Central America, are among the most species-rich cave communities in the world. The warm, humid conditions of tropical caves, combined with the abundant energy supplied by the guano of large bat colonies, support communities of beetles, flies, mites, cockroaches, and other invertebrates that can number in the hundreds of species in a single cave system. The Deer Cave in Sarawak, Malaysian Borneo is home to one of the largest bat colonies on Earth, with an estimated 3 million wrinkle-lipped bats roosting in the cave during the dry season. The guano deposited by this colony accumulates in deep layers on the cave floor, forming a substrate that supports a community of invertebrates of extraordinary diversity and abundance. The guano pile itself, a living and fermenting mass of bat droppings inhabited by countless organisms, is an ecosystem within an ecosystem, a biological entity that has been described by the researchers who have studied it as one of the most complex and least understood communities in tropical biology. The emergence of the Deer Cave bat colony at dusk is one of the most spectacular wildlife events in the world. 3 million bats streaming from a cave entrance in a continuous ribbon that can last for more than 2 hours, visible from kilometers away, attract predatory hawks and falcons that wait outside the cave entrance to pick off individual bats in the stream. Inside the cave, the smell, the sound, the heat generated by 3 million small warm-blooded bodies create conditions that are almost impossible to describe and that have no parallel in any surface environment. The ecological importance of this bat colony extends far beyond the cave itself. 3 million insectivorous bats consuming insects every night across the landscape surrounding the cave represent a predation pressure on insect populations that has a measurable effect on the composition and abundance of the insect community over a large area. Agricultural communities surrounding Deer Cave and similar large bat colonies have long recognized the value of bats as pest control agents. And research confirms that the presence of large bat colonies significantly reduces populations of agricultural pest insects in the surrounding landscape. The connection between cave-roosting bats and surface agriculture is one of the clearest examples of the ecological services provided by cave ecosystems to the human communities that live above them. A cave is not merely a geological feature or a biological curiosity. It is a component of the landscape with real and quantifiable effects on the ecology and economy of the surface world. Understanding these connections, mapping the flows of energy and matter and ecological influence that link the cave world to the surface world is one of the central challenges of modern cave ecology. It requires the integration of geology, hydrology, chemistry, and biology across spatial and temporal scales that exceed the scope of any single discipline. And it has been made more tractable in recent years by the development of new tools for studying underground environments. LiDAR, the laser-based three-dimensional mapping technology that has transformed the study of surface landscapes, has been adapted for use in caves, allowing researchers to produce three-dimensional maps of cave systems with centimeter precision without the years of surveying required by traditional cave mapping methods. These maps reveal not just the geometry of cave passages, but the spatial distribution of formations, water, sediment, and biological communities within the cave, providing a three-dimensional context for understanding the ecology of cave organisms that was previously impossible to obtain. Environmental DNA, the analysis of genetic material shed by organisms into their environment, has been adapted for cave waters and cave sediments, allowing researchers to detect the presence of species in cave systems without the need to physically observe or collect the organisms themselves. A water sample from a cave lake can now be analyzed to yield a list of the species present in that lake, including rare or cryptic species that would be missed by traditional sampling methods, providing a comprehensive picture of cave biodiversity that was previously unattainable. Stable isotope analysis, which uses the ratios of different isotopes of carbon, nitrogen, and other elements in organism tissues to trace the flow of energy through food webs, has been applied to cave communities to reveal the trophic structure of cave ecosystems in detail. The results have confirmed and quantified the dependence of cave communities on surface-derived organic matter, showing that even in cave systems far from any visible connection to the surface, the carbon in the bodies of cave organisms can be traced back to specific surface sources, demonstrating the hidden connectivity between the underground and the surface world. These new tools are transforming cave biology from a largely descriptive science focused on cataloging the extraordinary organisms that live in caves into a more mechanistic and predictive science capable of understanding and modeling the dynamics of cave ecosystems and their responses to environmental change. The transformation is ongoing and accelerating, driven by both the availability of new technology and by the growing recognition that cave ecosystems are not merely scientific curiosities, but are components of the global biological system whose health and function matter for reasons that extend far beyond their underground boundaries. The cave environments of the world contain what may be the most scientifically valuable collection of natural experiments in evolution available anywhere on Earth. The independence of cave colonization events, the completeness of the resulting adaptation, the availability of surface relatives for direct comparison, and the range of timescales across which cave evolution has operated. From a few thousand years in recently colonized systems to tens of millions of years in ancient isolated systems, make caves a uniquely powerful natural laboratory for studying the mechanisms of evolution. The insights generated by cave biology over the past century have been profound. The genetic architecture of complex adaptive traits, the mechanisms of sensory compensation, the life history consequences of energy limitation, the evolutionary dynamics of isolated populations, the molecular basis of convergent evolution. These insights have implications not just for our understanding of cave organisms, but for evolutionary biology as a whole, because the processes they reveal are the same processes that drive evolution everywhere, operating under conditions that make them more visible and more measurable than they typically are in surface ecosystems. But cave biology is not only about evolutionary mechanisms. It is also about the extraordinary organisms themselves, organisms that have been shaped by one of the most extreme and uncompromising environments on Earth into forms of beauty and complexity that have no parallel in the surface world. The limestone karst of southern China contains cave systems of a scale and antiquity that dwarf most of what exists elsewhere on the planet. The Guangxi and Guizhou provinces alone contain thousands of documented cave systems, many of them extending for dozens of kilometers through the interior of mountains that rise dramatically from the surrounding plain in the characteristic towers and pinnacles of karst topography, shapes formed over millions of years by the same dissolution processes that created the passages within them. The region has been inhabited by human beings for tens of thousands of years, and the caves have been known and used as water sources, shelters, and ceremonial spaces throughout that history. But the biological communities within them had been systematically studied for only a few decades, and what that study has revealed has consistently astonished the researchers conducting it. The cavefish of southern China represent one of the most diverse regional cave ichthyofaunas in the world. More than 100 species of cave-adapted fish have been described from Chinese caves, belonging to multiple families and representing independent colonization events from many different surface ancestors. The loaches, members of the family Cobitidae and several related families, have colonized Chinese caves, repeatedly producing dozens of blind, depigmented, cave-adapted species and cave systems distributed across the karst regions of Guangxi, Guizhou, Yunnan, and neighboring provinces. The cave loaches of the genus Troglodytes and the related genus Orionectes are among the most thoroughly studied, and their biology has illuminated aspects of cave adaptation that parallel and complement the findings from the Mexican Astyanax system. The parallel evolution of cave adaptations in Chinese and Mexican cave fish occurring in completely unrelated fish families in cave systems on opposite sides of the planet is one of the most compelling demonstrations of convergent evolution available anywhere in biology. The specific traits that have evolved in both systems: reduction of eyes, loss of pigment, elaboration of lateral line neuromasts, extension of lifespan, reduction of metabolic rate, these did not evolve because Chinese loaches and Mexican tetras share a common cave-dwelling ancestor. They evolved because caves present the same selective pressures everywhere. And because those selective pressures reliably produce the same evolutionary responses in vertebrate animals that have been adapting to them for sufficient time. The comparison between the two systems has gone beyond simply documenting the similarity of outcomes and has begun to probe the similarity or difference of the genetic mechanisms producing those outcomes. In Astyanax, the genetic architecture of eye reduction has been mapped in considerable detail, identifying specific genes and specific mutations responsible for the reduction of eye size and the degeneration of visual circuitry. The question of whether the same genes carrying similar mutations are responsible for eye reduction in Chinese cave loaches is being actively investigated, and preliminary results suggest a mixture of shared and divergent genetic mechanisms. Some of the same developmental genes appear to be involved in eye reduction in both systems, reflecting the constrained set of developmental pathways available for modifying eye development. But the specific mutations affecting those genes differ between the two fish groups, suggesting that while the developmental target of evolution is similar, the molecular path to that target varies across lineages. This pattern, shared developmental targets but different molecular mechanisms, is emerging as a general feature of convergent evolution across many biological systems. It suggests that convergence operates at the level of developmental processes and functional outcomes rather than at the level of specific molecular sequences, and that the space of evolutionary possibilities is constrained by developmental architecture in ways that are only beginning to be understood. The cave fish of China have also provided important insights into the ecology and behavior of cave fish that complement the extensive behavioral research conducted on Astyanax. The cave loaches of Guangxi have been studied in their natural cave habitats as well as in laboratory conditions, and this dual approach has revealed a nuanced picture of how cave fish use their sensory capabilities in the real cave environment. Cave loaches in their natural habitat show a pattern of space use that is strikingly different from what would be expected in surface fish of comparable size. They occupy specific microhabitats within the cave water body, often returning to the same precise locations repeatedly over periods of days, maintaining fidelity to specific current patterns, substrate types, and water chemistry conditions that appear to be associated with higher prey availability or lower predation risk, which requires the fish to navigate back to specific locations within a three-dimensional cave water body in complete darkness, demonstrates a spatial memory capability that has been confirmed by laboratory studies and that appears to be supported by the same kind of lateral line-based spatial mapping documented in Astyanax. The prey detection capabilities of the cave loaches have been measured in controlled experiments that allow the sensitivity of the lateral line system to be quantified. The results show that cave loaches can detect the water movements generated by a small invertebrate prey item at distances of up to 15 centimeters in still water and can track the position of moving prey with sufficient precision to orient a strike accurately from a distance of 10 centimeters. These capabilities exceed those of surface loaches by a factor of 3 to 5, representing a substantial functional enhancement of the lateral line system that is directly correlated with the elaboration of lateral line neuromasts documented in anatomical studies. The metabolic physiology of cavefish has been studied in detail in both the Astyanax and the Chinese loach systems, and the results reveal an adaptation to cave energy limitation that is more complex and more sophisticated than the simple reduction of metabolic rate that a naive account might predict. Cave fish do not simply have lower metabolic rates than their surface relatives. They have metabolic systems that are differentially tuned, with some processes running at lower rates than in surface fish and others running at comparable or even higher rates, depending on the functional requirements of each process in the cave environment. In particular, the metabolic processes associated with the development and maintenance of the lateral line system and the chemosensory systems are not reduced in cavefish. The energetic investment in non-visual sensory systems is maintained or enhanced, even as the investment in visual systems is reduced. This selective allocation of metabolic resources, reducing investment in useless structures while maintaining or increasing investment in useful ones, is the clearest possible demonstration that the cave syndrome is not simply the result of relaxed selection, allowing neutral deterioration of unused structures. It is the result of active selection for a specific allocation of limited energy resources that maximizes fitness in the cave environment. The distinction between neutral deterioration and active reallocation has been one of the most debated questions in cave biology for decades. The standard neutral model of eye loss in cave animals, which posits that eyes degenerate simply because mutations reducing eye development are neutral in darkness and accumulate by genetic drift without selection, makes specific predictions about the rate of eye loss and the genetic architecture of degenerate eyes that can be tested against the data. And the data, particularly from Astyanax, is increasingly inconsistent with the pure neutral model. The rate of eye loss in Astyanax cave populations is faster than the neutral model predicts for the observed mutation rate and population size. The genetic architecture of eye reduction involves not just random mutations affecting any component of the eye development system, but consistent changes in specific developmental pathways, particularly those involving the sonic hedgehog signaling pathway and its role in partitioning facial tissue between eye and jaw structures, and the correlation between eye reduction and enhanced taste bud development, which has now been demonstrated in multiple independent cave populations, strongly implies that selection is actively reducing eyes, because doing so provides a positive benefit in the form of enhanced chemoreception rather than simply because eyes are no longer useful. The emerging consensus in cave biology is that eye loss is driven by a combination of neutral mutation, accumulation in specific developmental pathways, and positive selection for the reallocation of that developmental resources, and that the relative contribution of these two processes varies among species, populations, and specific genetic pathways. This more nuanced model is consistent with the full body of evidence and makes predictions about the genetics of cave adaptation that are now being tested with the tools of modern genomics. The genomic analysis of cave animals has entered a new phase with the development of high-throughput sequencing technologies that allow the entire genomes of non-model organisms to be sequenced at a reasonable cost. Full genome sequences are now available for several cave animal species, including Astyanax mexicanus, the Proteus anguinus, and several cave beetles. And these sequences are providing an unprecedented view of the genetic architecture of cave adaptation. The genome of the published in 2020 by a team of researchers from Slovenia, France, and Germany, revealed several features of exceptional biological interest. The genome is extremely large, approximately 50 billion base pairs, more than 15 times the size of the human genome, a phenomenon called genome gigantism that has been documented in several amphibian groups and that appears to be related to the expansion of repetitive DNA elements rather than to an increase in the number of protein-coding genes. The biological significance of extreme genome size in amphibians is not fully understood, but it is associated with large cell sizes that may have consequences for the physiology and development of giant genome organisms. More relevant to cave adaptation, the genome shows signs of accelerated evolution in gene families associated with sensory function, particularly in genes involved in olfaction and in the detection of chemical gradients. The olfactory receptor gene family, which in most vertebrates is already one of the largest gene families in the genome, is unusually expanded in the olm, with a greater diversity of olfactory receptor genes than is seen in surface salamanders or in most other vertebrate groups studied. This expansion appears to reflect a diversification of chemical detection capabilities, that is, the genomic signature of an animal that navigates and hunts entirely by chemical sensing. The genome also contains evidence of gene loss in visual pathway genes, with several genes involved in phototransduction and retinal development, showing signs of pseudogenization, the accumulation of loss-of-function mutations that convert a functional gene into a non-functional relic. The pseudogenization of visual genes in the olm is consistent with the vestigial nature of its eyes and with the molecular story of vision loss that has been documented in other cave vertebrates. It also provides a molecular clock of sorts. The number of mutations accumulated in pseudogenized visual genes can be used to estimate the minimum time since the genes ceased to be under purifying selection, providing an independent estimate of the timing of the transition to cave life. For the olm, these molecular clock estimates suggest that the lineage has been adapting to cave conditions for at least 15 to 20 million years, consistent with geological evidence for the isolation of the dinaric karst cave systems in which olms live. This is one of the longest documented histories of vertebrate cave adaptation, and it explains the extreme degree of morphological and physiological specialization that the olm displays. 15 to 20 million years of cave evolution is a very long time for natural selection to work with. The cave invertebrates of the world's cave systems have received less genomic attention than the cave vertebrates, partly because many are too small or too rare to collect in sufficient quantities for traditional genomic methods, and partly because the phylogenetic context needed to interpret genomic data requires a well-developed understanding of the evolutionary relationships of cave species to each other and to their surface. Relatives, which in many invertebrate groups remains incomplete, but as sequencing technologies become cheaper and more sensitive, genomic studies of cave invertebrates are becoming more feasible, and the results are beginning to reveal aspects of cave invertebrate biology that were previously inaccessible. The cave beetles of the genus Anthroherpon in the caves of the Balkans have been the subject of genomic studies that have revealed a surprising degree of genetic differentiation between populations in different caves within the same cave system. Cave beetles that appear morphologically identical, that would be classified as the same species by traditional taxonomic methods, have been found to be genetically as distinct as recognized species in other invertebrate groups, representing what taxonomists call cryptic species, species that are genetically and presumably ecologically distinct, but that are not distinguishable by the morphological characters traditionally used to define species. The prevalence of cryptic species in cave faunas is now recognized as one of the most significant findings of the genomic era of cave biology. Systematic surveys of cave invertebrate diversity using molecular methods have consistently found 2 to 5 times more species than had been estimated by morphological taxonomy alone. This means that the global diversity of cave life is substantially greater than previously thought, and that the conservation assessments based on morphological species counts significantly underestimate the number of distinct evolutionary lineages at risk in cave ecosystems. The discovery of cryptic species in caves has practical as well as scientific importance. Conservation strategies for cave species must be designed at the level of the management unit, the population or group of populations that is genetically and demographically independent from other populations, and that must be managed separately to maintain its evolutionary trajectory. a cryptic species that occupies a single cave system, and that is not recognized as distinct from its neighbors by morphological taxonomy, may be omitted from conservation plans based on those taxonomic assessments, and may go extinct without ever having been recognized as a distinct entity. This is not a hypothetical scenario. Molecular surveys of cave systems in Europe, North America, and Australia have repeatedly identified cryptic species that have not been described by traditional taxonomy, including species that are represented by populations of only a few hundred individuals in a single small cave system. Some of these cryptic species have been discovered in cave systems that are already heavily impacted by human activities, raising the possibility that extinction has already occurred in some cases before the species in question was even known to exist. The relationship between cave biology and the human communities that live above cave systems is a complex one that has evolved substantially over the past century. For most of human history, caves were used primarily as sources of water, as shelters, and as spiritual spaces, with little recognition of the unique biological communities they contained. The systematic study of cave biology began in the 19th century in Europe, initially driven by the discovery of the ulmen and by the taxonomic work of early spelunkers who collected cave invertebrates as curiosities of natural history. The development of cave biology as a rigorous scientific discipline accelerated in the 20th century, driven by the development of modern speleological techniques that made deep cave exploration safe and systematic, by the development of ecological and evolutionary theory that gave the phenomena observed in caves a theoretical context, and by the growing recognition among biologists of the unique scientific value of cave environments as natural experiments in evolution. The practical connection between caves and human welfare is mediated primarily through groundwater. The majority of the world's cave systems are formed in aquifer rocks, limestone, dolomite, and gypsum that also serve as important sources of drinking water and irrigation water for human communities. The Dinaric karst of southeastern Europe supplies drinking water to millions of people in Slovenia, Croatia, Bosnia-Herzegovina, Albania, and Montenegro. The Edwards Aquifer of Texas, formed in karst limestone, supplies water to more than 2 million people in San Antonio and surrounding communities. The karst aquifers of the Yucatan Peninsula supply water to millions of residents and tens of millions of tourists in the region. The water that flows through these aquifers does not pass through a simple pipe from surface to tap. It moves through a complex three-dimensional network of fractures, passages, and pores that collectively constitute the cave environment, and it is in contact at every point of its journey with the biological communities that inhabit that environment. The bacteria that live in cave biofilms, the invertebrates that feed on organic matter carried by cave water, the biological processes of decomposition and mineral transformation that characterize cave ecosystems. All of these contribute to the physical and chemical quality of the water that emerges at springs and wells. The relationship is bidirectional. Cave ecosystems depend on the quality of the water that enters them from the surface, and the quality of the water that emerges from caves for human use is partly determined by the biological processes occurring within them. This mutual dependence makes the health of cave ecosystems a matter of direct human interest, not just scientific interest, because the degradation of cave ecosystems through contamination or the disruption of cave biological communities can have measurable effects on the quality of groundwater that depends on those communities for its biological purification. The karst aquifer systems that supply drinking water to large human populations are also among the most vulnerable to contamination, unlike consolidated rock aquifers. where water moves slowly through tiny pores and is effectively filtered of contaminants by the long residence time in the rock. Karst aquifers transmit water rapidly through large passages with little filtration. A contaminant introduced at the surface of a karst landscape can reach a spring or well kilometers away within hours, bypassing the natural filtration processes that would degrade the contaminant in other aquifer types. This vulnerability to contamination has been demonstrated repeatedly by pollution incidents involving karst aquifers worldwide. Agricultural pesticides and fertilizers applied to fields above karst aquifers have repeatedly appeared in springs and municipal water supplies at concentrations requiring treatment. Industrial solvents and fuel spilled on karst surfaces have been traced to springs many kilometers away within days of the spill. The rapidity of contaminant transport through karst systems reflects the same hydraulic connectivity that makes caves biologically rich. The large passages that allow cave organisms to colonize new habitats and that allow organic matter to flow from the surface to the cave interior also allow contaminants to flow rapidly from the surface to the water supply. The protection of karst groundwater resources therefore requires the protection of the entire surface area that contributes water to the cave system, which in large karst aquifers can extend over hundreds or thousands of square kilometers. This is a formidable land management challenge that has been addressed with varying degrees of success in different jurisdictions. In Slovenia, the protection of the Postojna planina cave system and its associated groundwater resources is embedded in a legal and planning framework that regulates land use across the entire catchment area of the cave system, reflecting the recognition that the cave and its surface catchment are a single hydrological and biological unit that must be managed as such. The Postojna cave system itself is one of the most biologically significant and most visited cave systems in the world. More than 30 species of cave-adapted invertebrates have been described from Postojna, including the olm, which has been known from Postojna since the 18th century, and which was the first cave animal to be subjected to systematic scientific study. The cave has been open to tourists since the early 19th century and now receives more than 500,000 visitors per year, making it one of the most visited tourist attractions in Europe. The management of tourist visitation in a biologically sensitive cave system presents challenges that have no perfect solution. The presence of large numbers of visitors introduces non-native organisms, including bacteria and fungi on clothing and equipment, modifies the cave's air circulation patterns through the opening and closing of doors and the body heat of visitors, alters the deposition patterns of cave formations through the introduction of nutrients that support algal growth on illuminated surfaces, and disturbs the cave fauna through noise, vibration, and the physical impact of visitors on cave surfaces. At Postojna, these impacts have been managed through a combination of physical infrastructure, controlled visitor routing, lighting systems designed to minimize photosynthetic algae growth, and monitoring programs that track the condition of cave formations and the status of cave biological communities over time. The program is imperfect. Some algal growth on illuminated surfaces is inevitable, and the thermal and chemical modifications of the cave atmosphere caused by large numbers of visitors cannot be entirely eliminated. But the combination of management measures has allowed the cave to accommodate massive visitor numbers while maintaining the biological community that makes it scientifically valuable. The tension between the scientific and conservation value of caves and their economic potential as tourist attractions is a tension that cave managers around the world navigate with varying levels of success. The economic value of cave tourism is real and significant. The direct revenue from cave admission fees and the indirect economic activity generated by tourism in the regions surrounding major show caves contribute substantially to local and regional economies. In countries and regions where alternative economic development opportunities are limited, this revenue can be the difference between economic viability and poverty for rural communities. The conservation case for managing tourist caves carefully is equally compelling. A cave ecosystem that has been degraded by unmanaged tourism cannot be restored on any timescale relevant to the communities that depend on it. The cave formations that draw tourists, the stalactites and stalagmites and flowstones and cave pearls, grow at rates of millimeters per century, and a formation that is broken by a careless visitor will not be replaced in any human lifetime. The cave organisms that make these systems scientifically valuable are often so specialized and so rare that a single contamination event or habitat disturbance can eliminate a population that has been evolving in its specific cave environment for millions of years. The balance between use and conservation in cave tourism is therefore one of the most important and most difficult problems in cave management, and it will not be solved by a formula that applies equally to all caves and all situations. It requires site-specific knowledge of the biological and physical sensitivity of each cave, careful assessment of the type and magnitude of human impacts associated with different kinds of visitation, and the development of management regimes that are both biologically sound and economically viable. What is certain is that the scientific and conservation value of cave ecosystems will continue to grow as the biological richness of these environments becomes better understood, as the evolutionary insights they provide become more widely appreciated, and as the connections between cave ecosystems and the human communities that depend on cave groundwater resources become more clearly established. The invisible world beneath our feet is not a peripheral curiosity. It is a central component of the planet's biological heritage and of the hydrological systems on which human civilization depends. The researchers who study this world, the cave biologists, hydrogeologists, speleologists, and evolutionary biologists, who spend their careers in the dark, mapping passages and collecting samples and assembling the extraordinary biological and geological story of the underground, are among the most dedicated and least recognized scientists in any field. They work in conditions of physical difficulty and occasional genuine danger. They study organisms that are invisible to the general public and that lack the charismatic appeal of lions or whales or eagles. They make discoveries of profound scientific significance that receive a fraction of the media attention devoted to more glamorous fields. But the knowledge they are accumulating, discovery by discovery and cave by cave, is indispensable. It is knowledge about how life works at its most fundamental level, about the mechanisms of evolution operating under conditions that strip away the complexity of surface ecosystems and reveal the basic logic of natural selection with unusual clarity. It is knowledge about the history of the Earth, recorded in the chemistry of cave formations over timescales that no other archive can match. It is knowledge about the resources that human civilization depends on, the groundwater that flows through caves before it reaches our wells and our rivers and our taps. And it is knowledge about the extraordinary variety of forms that life can take when evolution has sufficient time, sufficient isolation, and the specific selective pressures of permanent darkness. The creatures of the cave worlds are not marginal. They are not vestigial remnants of surface life that failed to leave when the conditions changed. They are fully realized, completely adapted, deeply successful inhabitants of a world that runs on different rules than the one we inhabit. They have been in the dark far longer than we have been in the light, and they have made of that darkness something that we are only now beginning, with the limited tools of a species that has been scientifically curious for only a tiny fraction of the time that cave life has been evolving, to understand. The understanding, when it comes in full, will change how we think about life itself, about what it needs, about what it can do without, about the range of conditions under which the chemistry of evolution can produce minds, senses, strategies, and adaptations of a complexity and precision that rivals anything the sunlit world has produced. We are at the beginning of that understanding. The caves are deep. The organisms within them are many, and most are unknown. The science is young relative to the antiquity of its subject, but the direction is clear. Every expedition that returns from a cave system with new specimens and new data adds to a picture that is becoming more astonishing with each edition. Every genome sequenced from a cave organism reveals mechanisms of adaptation that were invisible before the tools existed to see them. Every speleothem record extending the climate archive into a new period of Earth history adds a data point to a record that connects the underground world to the history of the entire planet. The cave worlds are not separate from the world we live in. They are beneath it, connected to it by geology and hydrology and evolutionary history, shaping it in ways we are still learning to measure. Populated by forms of life that reflect the same processes that produced us, operating under conditions that reveal those processes with an unusual and invaluable clarity. Go deep enough into any of the world's great cave systems, deep enough that the last trace of surface light has been left behind and the temperature has stabilized to its constant year-round value, and the silence is as complete as silence ever gets. And you are in a place that has not changed in its fundamental character for millions of years. The darkness is not empty. The silence is not dead. The cold is not sterile. Something is moving in the water. Something is hunting in the passages. Something is growing slowly on the cave wall, deriving energy from chemistry that has nothing to do with the sun. Something has been doing these things in this place since before the mountain above you existed in its current form, since before the mammals that would eventually produce your ancestors had appeared, since before the flowering plants had diversified across the surface of the world, and it is doing them still. The deep caves of the Atacama Desert in Chile Harbor, a community of microorganisms that pushes the boundaries of what we thought life could do. The Atacama is the driest non-polar desert on Earth, receiving on average less than 15 millimeters of rain per year, and in some areas of its hyper-arid core, no measurable precipitation has fallen in recorded history. The surface of the Atacama is as close to lifeless as any place on the land surface of the Earth. But beneath the surface, in the cave systems formed by evaporite dissolution, in the salt-rich soils and rocks of the Atacama, and in the rock itself, in tiny pores and cracks too small to see with the naked eye, there are microorganisms. They metabolize at rates so slow that they may divide only once every 1,000 years. They derive their energy from the radioactive decay of minerals in the surrounding rock, using the radiation to split water molecules and produce hydrogen that serves as an energy source. They have not seen the surface in millions of years. These organisms are relevant to a question that has become one of the most important in all of science. Is there life elsewhere in the solar system? Environments of permanent darkness, extreme cold or heat, complete absence of surface-derived organic matter, and total isolation from the sunlit biosphere are not unique to Earth. They are replicated with varying specifics on bodies throughout the solar system. Europa, the moon of Jupiter, has a surface covered in ice and an ocean of liquid water beneath that ice, heated by tidal flexing from Jupiter's enormous gravitational field. That subsurface ocean is in contact with a rocky seafloor that is almost certainly geologically active, producing hydrothermal vents that could support chemosynthetic communities similar to those of Earth's deep-sea vents, and perhaps similar to the chemosynthetic cave ecosystem of Mobile Cave. The subsurface ocean of Europa is sealed from space by kilometers of ice. It has no sunlight. It has no surface-derived organic matter. It is, in the relevant biological terms, an enormous cave. Enceladus, the moon of Saturn, has an even more dramatic subsurface ocean, one that is actively venting through geysers in the South Polar region, shooting plumes of water ice and organic molecules into space where they can be sampled by passing spacecraft. The Cassini spacecraft, which orbited Saturn from 2004 to 2017, flew through these plumes and detected water, hydrogen, carbon CO type, and a range of organic molecules, including simple amino acids, the building blocks of proteins. The chemistry of the Enceladus plumes is consistent with hydrothermal reactions in the rocky seafloor of the subsurface ocean. Those reactions, occurring in the complete absence of sunlight at the bottom of an ocean sealed beneath many kilometers of ice, are exactly the kind of reactions that support chemosynthetic communities in Earth's deep-sea hydrothermal vents and in chemosynthetically based cave systems like Mobile. Mars has a network of cave systems that was formed by the same lava tube processes that create the lava tube caves of Hawaii and the Canary Islands. These Martian caves, visible as sinuous collapse features on the surface, extend for hundreds of kilometers and reach depths of many tens of meters below the Martian surface. The Martian surface today is sterilized by ultraviolet radiation, stripped by the thin atmosphere and by ionizing cosmic radiation, cold and desiccated. But the interior of Martian caves is shielded from these surface hazards, maintains temperatures several tens of degrees warmer than the surface, and may retain liquid water at depth from the geothermal activity that was certainly more intense in Mars's past. If life ever existed on Mars, the caves of Mars are among the most likely places for it to have survived into the present. In the same way that cave microorganisms on Earth survive in environments where surface life cannot persist. The study of cave biology on Earth is therefore not just the study of an unusual biological niche. It is the preparation for the search for life in places where surface conditions are inhospitable, but cave conditions may be survivable. Every chemosynthetic cave ecosystem discovered on Earth refines our understanding of the minimum conditions required for complex biological communities to exist. Every cave microorganism adapted to extreme cold, low energy, and isolation expands our understanding of the range of conditions that life can tolerate. Every metabolic strategy discovered in cave organisms represents a potential metabolic strategy for life in the subsurface environments of other worlds. The astrobiology community has recognized this connection, and cave biology has become increasingly integrated into the research programs of space agencies studying the potential for life elsewhere in the solar system. NASA's Subsea program, the Systematic Underwater Biogeochemical Science and Exploration Analog, studies deep-sea hydrothermal environments as analogs for Europa's subsurface ocean. The caves of the Atacama, the lava tube caves of Hawaii, and the gypsum caves of the Canary Islands are now regularly studied as analogs for the potential cave environments of Mars and other bodies. The microbial communities in these cave environments are characterized in detail, their metabolic strategies documented, their genomic capabilities sequenced to build a database of biological strategies for surviving in cave-like conditions that can inform the search for life beyond Earth. The profound scientific importance of cave biology coexists with a relatively low public profile, a gap between scientific significance and public awareness that has real consequences for conservation and funding. Surface biodiversity, the charismatic megafauna of forests and grasslands and reefs, attracts public attention and conservation funding in ways that the invisible fauna of underground environments. This is beginning to change, driven in part by the same technological advances that are transforming cave biology scientifically. High-quality underwater cave photography and videography have made the visual world of cave diving accessible to audiences who will never experience it directly, revealing the extraordinary aesthetic beauty of underwater cave environments, with their stalactites and stalagmites reflected in the mirror-calm surfaces of cave lakes and their ghostly white fauna drifting through the clear, cold water. The cave diving community has been a critical partner in cave biology research, providing the access to underwater cave environments that biologists could not achieve alone, and contributing observations and samples from cave systems that would otherwise remain unstudied. Cave divers are among the most technically skilled and personally courageous practitioners of any sport or science, operating in environments where an equipment failure or a navigation error can be fatal, with no possibility of rescue and no margin for error. The exploration of underwater cave systems by cave divers has repeatedly revealed biological communities of extraordinary scientific interest. The Yucatan Peninsula of Mexico contains one of the most extensive underwater cave systems on Earth, the Sac-Acton system and its connected passages. Formed in limestone during periods when sea levels were lower and the caves were air-filled and subsequently flooded as sea levels rose at the end of the last ice age. These flooded passages connect the interior of the Yucatan to the Caribbean Sea, forming a complex hydrological system that has been explored by cave divers over several decades. The biological communities in the Yucatan cave systems include organisms of exceptional interest, blind and depigmented cavefish of the genus Typhliacina, cave shrimp of the genus Typhlotia, that have been genetically shown to be more closely related to species in the caves of Cuba and Puerto Rico than to species on the Yucatan surface, reflecting a history of colonization and dispersal that occurred when the land areas of the Caribbean were connected by dry land during the lower sea levels of glacial periods. The Yucatan cave shrimp demonstrate a principle of biogeography that is increasingly recognized as important for understanding the distribution of cave fauna globally. Caves are not isolated biological islands, but are connected to each other through underground hydrological systems, and species have dispersed through these systems in ways that reflect the geological history of the connected cave systems rather than the surface geography of the overlying landscape. Species found in cave systems, thousands of kilometers apart, may be more closely related to each other than to species on the surface directly above them, because they are connected by underground routes that have no surface expression. The Tulum cave system in the Yucatan, one of the most intensively explored underwater cave systems in the world, also contains one of the most significant paleontological records in the Americas. Human remains, stone tools, and the bones of extinct megafauna, including giant sloths, gomphotherds, and tapirs, have been found in the air pockets of the Tulum caves. deposited when the caves were dry during the last ice age and subsequently sealed and preserved by the flooding that occurred as sea levels rose. These remains include some of the oldest human skeletal material in the Americas, dating to more than 13,000 years before present, providing evidence of human occupation of the Yucatan Peninsula that predates most other sites in North America. The caves of the world are not only living biological laboratories, they are archives of deep time. The sediments that accumulate in cave floors record millennia of environmental history with a completeness and precision that surface sediments rarely achieve. Speleothems, the collective name for the mineral formations, stalactites, stalagmites, flowstones, and cave pearls that form in caves by the deposition of minerals from percolating groundwater are among the most important natural archives of past climate available to science. The chemistry of speleothem growth layers records the chemistry of the water that deposited them, which in turn reflects the temperature, vegetation cover, and precipitation patterns of the surface above the cave at the time of deposition. By measuring the isotopic ratios of oxygen and carbon in speleothem growth layers, scientists can reconstruct the climate of the past with a temporal resolution of years to decades over time spans of hundreds of thousands of years, providing records of climate change that complement and extend the ice core records from Greenland and Antarctica. The Cave of Villers in France contains speleothems that record the climate of southwestern Europe over the past 450,000 years, including multiple cycles of glacial advance and retreat, the warm interglacial periods between ice ages, and the abrupt climatic oscillations that characterized the last deglaciation. The Cave of Heishang in China contains a speleothem record of the East Asian monsoon that extends back 350,000 years, providing one of the longest continuous records of monsoon variability in existence. These climate records from caves have been essential for understanding the mechanisms of past climate change and for testing the climate models used to project future climate under different greenhouse gas emission scenarios. A climate model that cannot reproduce the patterns of past climate change recorded in cave speleothems is a model with significant weaknesses. The calibration of climate models against speleothem records has been one of the most important contributions of cave science to the understanding of climate dynamics. The archaeological record preserved in caves is no less significant. Caves have been used by human beings as shelters, as ritual spaces, as burial sites, and as galleries for art since the earliest periods of human prehistory. The cave art of Western Europe, produced by anatomically modern humans between 45,000 and 10,000 years ago, represents the earliest unambiguous evidence of symbolic thought in the human record. The capacity to create representations of the world that are not the world itself, to use marks and pigments to convey meaning across time and space. The cave art of Lascaux in France, Altamira in Spain, Chauvet in southern France, and dozens of other sites in Europe, Africa, Asia, and Australia reveals the minds of our ancestors with an immediacy that no other record from the distant past can match. The horses and bison and mammoths depicted on cave walls with such extraordinary skill and such evident understanding of animal anatomy and movement were painted by people who were biologically indistinguishable from us, whose brains were as complex and as capable as ours, who looked at the world with eyes identical to our own and found in it the same things that we find in it: beauty, power, danger, mystery. The choice of caves as the location for this art was not arbitrary. Cave art is concentrated in the deepest and most inaccessible parts of cave systems, in chambers that could only be reached by crawling through narrow passages with torches that produced unreliable light and that consumed the limited oxygen of the cave atmosphere. The people who made this art did not paint in caves because caves were convenient. They painted there because caves were significant, because the darkness and the depth and the physical difficulty of reaching the underground world gave it a meaning that surface locations did not have. What that meaning was, we cannot know with certainty, but the universality of the association between caves and the sacred across human cultures from the Paleolithic to the present suggests that something about the cave environment, its darkness, its silence, its otherness, its sense of being a space outside the normal world, has consistently evoked responses in the human nervous system that are associated with heightened significance, with the sense of crossing a threshold between the ordinary and the extraordinary. That sense is not misplaced. The underground world is extraordinary. It is a world where the rules of the surface, the dependence on sunlight, the organization of life around the daily cycle of light and dark, the competition for space and resources in full view of predators and rivals do not apply. It is a world where evolution has been working in different directions and under different constraints than anywhere on the surface, producing forms of life that illuminate by their very difference the processes that have produced all life everywhere. The creatures of the cave worlds, the blind fish with their extraordinarily sensitive lateral lines, the white salamanders that live for a century in the dark, the bacteria that build ecosystems from the chemical energy of rock, the glowworms that manufacture light where no light exists, the cave spiders that taste the air with antennae fine-tuned beyond what any surface spider can achieve, are not failed versions of their surface relatives. They are perfected versions of the cave's demands. They are what 100 million years of underground evolution produces when it has the right starting material and the time to work. They are also, each of them, a question addressed to the surface world, a question about what we take for granted about the structures and senses and capabilities that we and our surface relatives depend on, which these animals have shown are not necessities, but contingencies, not universal features of life, but specific adaptations to a specific environment that is not the only environment possible. The blind cavefish does not miss its eyes. The white salamander does not experience its lack of pigment as a deprivation. The bacterium deriving energy from rock chemistry is not aware of the sunlight it does not need. They are complete. They are sufficient. They are exactly what their world has made them. And their world is as real and as full and as biologically rich as any place that daylight touches. What we learn from them as we continue to study them is not simply the detailed biology of cave organisms, though that biology is extraordinary in its own right. What we learn is something about the nature of adaptation itself, about the way natural selection responds to the specific pressures of specific environments with a creativity that consistently exceeds what we could have predicted from first principles, about the way evolution finds solutions to problems we did not know were problems, about the way life, given sufficient time and a consistent set of selective pressures, transforms itself into something so precisely fitted to its circumstances that the transformation looks, in retrospect, inevitable. It was not inevitable. It never is. Evolution does not have a destination. It does not move toward a goal. It responds generation by generation to the specific conditions it faces, accumulating changes that each make their carrier slightly more likely to survive and reproduce in those conditions, until the accumulated changes add up to something that looks like a design but is not, something that looks like a plan but is not, something that looks like inevitability but is, in its truest nature, nothing more and nothing less than the blind, patient, unimaginably powerful accumulation of successful accidents over time. The cave worlds teach us this. They teach it with a clarity that surface environments, where the complexity of ecological interactions obscures the direct line between selective pressure and adaptive response, cannot always achieve. The pressure in a cave is simple or simpler. Darkness, cold, scarcity. The responses are precise and precisely readable. The mechanism is visible, and the result, the animal that navigates the dark with senses tuned beyond anything that sunlight could have demanded, the organism that builds a living ecosystem from the chemistry of rock, the creature that illuminates its own small patch of the underground with light manufactured from its own biochemistry, is as close to a demonstration of the power of evolution as any place on Earth can offer. The caves are dark, and in the dark, extraordinary things have learned to live.